Greetings and welcome to the final Cold Regions Research Center speaker event of this academic year, Wildlife Health and Surveillance in a Changing Arctic with Dr. Suzanne Kutz. My name is Alex Latta. I'm a faculty member here at Laurier and also interim director of the Cold Regions Research Center. I'd like to extend thanks to the Laurier Center for the Study of Canada for co-sponsoring the event and for hosting us on their webinar platform. Thanks also to Gifty Atya for her work working with our speaker to arrange the talk, and to Trina King for her work on promotional materials. Dr. Kutz will present for about 45 minutes, after which we'll have about 15 to 20 minutes for questions. And before we start, I'd like to alert you to the fact that we are recording the event. As long as all goes well, we will post it later to our website. One other housekeeping note is around audience participation. You will be able to pose questions to the speaker by using the question and answer function at the bottom of your screen. And we do encourage you to post questions as they occur to you so that we have some ready to go when we arrive at the end of the talk. Before we introduce our speaker, I'd like to offer a brief land acknowledgement. Indeed, let us all acknowledge the numerous Indigenous peoples whose lands we are calling in from today. We're hosting here at Laurier from the traditional territories of the Arawandran, Anishinaabe, and Haudenosaunee peoples. For our guests from outside the region, I will share that Laurier sits on a 950,000 acre tract of land. That's 6 miles on either side of the Grand River, which was granted to the Haudenosaunee Confederacy in 1784 by the British after the Haudenosaunee fought on the side of the British in the American Revolutionary War. Subsequently, that land has been eroded to just 5% of its former extent, where the Six Nations Reserve currently sits. Despite the details specific to this region, it is a familiar story of dispossession through the imposition of Crown authority and then private ownership of land, underwritten since 1867 by the powerful fiction of Canadian sovereignty. This territory is also governed by the One Dish, One Spoon Treaty between the Haudenosaunee and Anishinaabe confederacies. That treaty signals that the wealth of the land is to be shared and cared for in common, a commitment that we would all do well to take up in the ways that we live our lives and do our work and relate to one another. The existence of this treaty is also a good reminder that settler society governments were latecomers to the work of environmental governance and conservation here on Turtle Island. I'd like to further acknowledge that for those of us who are researchers or workers in environmental change, conservation, sustainability, and related fields, we owe a debt of gratitude to the lands and peoples where we are guests as we do our work. With this gratitude also comes responsibility to advance that work in ways that honor and support Indigenous stewardship practices, which build on millennia of cohabitation and relationship between humans and non-human others. I think our talk today is going to give us some good food for thought in this regard. To introduce our speaker and to take on the role of moderating questions and closing the event, I welcome Dr. Frances Stewart to the virtual podium. Dr. Stewart is a faculty member and researcher in the Department of Biology at Laurier, where she leads the Wild Lab. She and her team of students conduct research on the responses of wildlife to natural and human-caused environmental change, with that work currently focused in the Northwest Territories and Northern Ontario. Over to you, Dr. Stewart.
Thank you very much for that, Alex. That was fantastic. It is my pleasure today to be introducing Dr. Susan Kutz. So Susan is a professor at the University of Calgary, Faculty of Veterinary Medicine, a Canada Research Chair in Arctic One Health, and a Fellow of the Canadian Academy of Health Sciences. She's worked in the Arctic since 1988, addressing health at the environment, animal, and human interface. In partnership with Indigenous communities, she initiated and maintains community-based wildlife health surveillance programs with the goal of detecting early changes in wildlife populations, protection of public health, and facilitating the incorporation of local and traditional knowledge into wildlife co-management. Bringing together local, traditional, and scientific knowledge is a key theme throughout her work. So, for example, she pioneered the Northern Community Health Rotation in NWT Satu settlement area, where she and her team have delivered annual veterinary services to 5 Dene communities since 2009. In addition to providing essential animal health services, this program provides unique cross-cultural experiential learning opportunities for veterinary students and mentorship in the health sciences for Indigenous youth as well. It's a privilege to have you here with us today, Susan. And without further ado, I will give it over to you for your webinar. Thank you.
Well, thank you very much for the nice introduction, and it's a real pleasure to be here and a real honor to be able to give this talk today. I am coming to you from the land of Treaty 7 peoples and work up in the Arctic across a variety of different areas. So today I'm going to talk about wildlife health and surveillance in a changing Arctic. And I see a number of people on the participants list here whose names I recognize, and many of whom are from the north. So a real pleasure to have you all here. Before I go forward, I just want to acknowledge that what I'm presenting today is the result of multitudes of graduate students, postdocs, summer students, and colleagues at work, as well as really close partnerships with the communities of Ekaluktuchuk or Cambridge Bay, Koluktok, and Uluhokto, as well as governments of Northwest Territories and Nunavut. And so while I present it, it's certainly the work of many, many people. I also want to acknowledge the many mentors that I've had throughout the years working in the north, in the Sahtu, and in the Kitikmeot and in Nunavut regions of the Nunavut and the Northwest Territories. Again, I've learned, I think, more than I've given up north, and it's always a growing experience. So I'm going to start my talk with this picture, and this is a picture I took on Ellesmere Island in 2021. A beautiful scene, a beautiful environment, but a really unfortunate event where we've got a dead muskox here. And the reason I've got this picture is because I got this text from the tundra late on August 16th, essentially saying to me, hello, Dr. Kutz, I'm writing from Ellesmere Island. Dean Clough, who's actually on the call right now, gave me your contact as I found 20 plus dead muskoxen on the north end of Fosheim Peninsula. So this was quite alarming, and I followed up and asked Ronan a little bit more about this, and he and his team had been up there since about the third week of July. They were a film crew filming wolves. They had recorded mortality sites of over 20 muskoxen that had died between August 3rd and 16th. And at the time, he said the weekly valley population count was about 150 to 160 animals. I worked with various different people up north to try and figure out how on earth we can investigate this. It's a very remote area. This is the Google screen capture of trying to get from Calgary International Airport to Eureka, which is on the Fosheim Peninsula, and there are no flights. So what we were able to do, again, because of a number of different people who were linked into this, we were able to find out that there was a resupply charter leaving in one day from Yellowknife, and it was going to be going up to Eureka. Cost to get up there, of course, extremely expensive, about $10,000 round trip with the flights only. But we were able to make this happen because of a number of different people. So I went up to Yellowknife, up to Resolute Bay, picked up a wildlife officer who came out to Eureka with me and helped out. We flew out there, spectacular scenery, and arrived on Friday night at about 5 o'clock in Eureka. This is the end of the runway in Eureka. There, Environment Canada Weather Station had put a couple quads at the end of the runway for us to take, and the film crew had come in from their field camp out at Eastwind Lake and picked us up. So we borrowed the quads, we drove out to this location and started our investigation. Of course, not without a little bit of trouble from the wolves. So again, this was about 2.5 day preparation to get up here to this very remote area to try and investigate this really alarming outbreak of mortalities, of muskox mortalities in a really short period of time. So we loaded up our field gear onto the quads and went out and started to investigate these dead muskoxen. We took samples, locations, all the information we could get. We were geared up in reasonably good personal protective gear because we didn't know what was causing these mortalities. And of course, anything that causes things to die quickly is often something that can also cause people to die quickly. So we also had to be really aware of these precautions. Over the course of 2.5 days, we were able to get to 10 of these carcasses. It became obviously very depressing as one goes from one carcass to the other, wondering what's going on. And then after that, got flown out of Eureka back to Resolute Bay with a short stop at a field camp on another island, made it to Resolute, and in 3 days' time, I was back to Calgary. Unfortunately, my supplies, my coolers full of samples ended up split between Resolute Bay, Iqaluit, and Ottawa, and they didn't arrive in Calgary until September 15th. So I tell you all of this just to emphasize the challenges of trying to get any sort of investigation done on wildlife mortalities or morbidity, sicknesses in the Arctic. This was a kidney. You can see it's just mush. And this is a joint, the fault of both us not being able to get fresh carcasses when the mortality event happened, and also probably some less than ideal handling when it was being shipped to Calgary and the delays that were required. So what this meant is that up here you can see the pathologist's report. Tissues were markedly autolyzed, making histological evaluation of minimal value. There were no significant findings. Ancillary testing will depend on findings in other members of this cohort. So essentially the histology was useless as far as trying to find out why these animals died. Fortunately, we did have a number of samples from many different animals, and we were able to culture from them. And despite the really horrible condition they were in, given the presentation that we saw, which was rapid death of animals of all ages, good body condition, suggesting it was a septicemia. But given that and the consistent culture of the bacteria Erysipelathrix ruseopathy from all tissues tested, we had a tentative diagnosis that this bacterium was the one that was causing the mortality. So this is all to give you some context of the North, and I think many of the participants on this call right now have been in the North and they understand some of these challenges. And while we're not likely to have the next pandemic emerge from the Arctic, nevertheless, emerging infectious diseases are really, really quite important there and represent a number of different challenges to the northern environment and northern peoples. First and foremost, it is remote and sparsely populated. And so that means when you get some of these big mortality events or health crises, it's very hard first to detect them. The only reason we knew about this, and to this day, the only reason we know about this die-off was because there was a film crew there at the time. So that these vast areas mean that there's very poor detection to mortality events. And the challenges of responding to them are also immense, as you can see from just this simple trip that I did up there. In addition, when these things when you get an outbreak like this and it's in humans, there's also very limited medical resources. And often the health centers are limited to just trying to treat the sign they see, but they may not be able to delve deeper into understanding what the disease is. We're also dealing with an area where the rapid rate of climate and other change is very conducive to disease emergence, and I'll speak more about that, and also highly vulnerable populations where the impact of disease is disproportionately great because of poverty, food insecurity, and also the conservation and cultural aspects. I have to apologize. I have two puppies that are playing pretty hard right now. The challenges of giving a presentation from home. So climate change in the Arctic, as we know, is happening very rapidly, more rapidly than anywhere else in the world. And this is altering these Arctic ecosystems. So on the left, you see this classic classic figure of how warming is happening more rapidly in the Arctic regions. And on the right, you see the extent of sea ice. And I want to touch on this sea ice, the changes in sea ice, because several years ago, Post et al. Put out a really nice article in Science talking about how sea ice changes are affecting the ecology of the North. And I'll link this to emergence of infectious disease. So this is a figure from this article that they wrote in 2013, just showing you what the world looked like in the 20th century. So, what the Arctic world looked like in the 20th century. There was lots of sea ice. It served as a platform for pupping, for walruses to go down and get benthic invertebrates. It was very lush along these edge of the sea ice. Belugas would hang out around it. Polar bears use it. Arctic fox would use it as a corridor to travel around the Arctic and across Arctic islands. These ice on the land would be, and ice patches on the land would be cover or insect or leaf habitat for caribou. And at the same time, that sea ice also acted as a barrier for other species coming up, such as killer whales. So this was what we would see back in the 20th century. Fast forward to today, and we see these massive changes that are really dramatically altering the ecology of not only the marine environment, but the terrestrial environment. And here you see, without the sea ice platform, there's not nearly as, the ice edge is much smaller, it's not as rich. We see things like walruses having to go to the land instead of using the sea ice for as a platform, and not being able to make it out to their desired food sources, causing energetic costs. We see stress and disturbance because of ships and increasing tourism with tourism and cruise ships. We see that now the sea ice that was once a barrier to animals like killer whales is now a corridor for killer whales, yet the sea ice absence is now a barrier to things like Arctic fox that would have been using that for extended periods of times. Phenological mismatch with earlier plant growth. We no longer have as much snow and ice on the mainland, so less insect relief for the animals, thus also increasing stress. We get crowding because of limited amount of haulouts and increased opportunities for disease transmission. And then relaxation of temperature constraints on parasites. So with the reduction of sea ice in the marine environment, it also has profound effects on the terrestrial environments. And this really becomes the perfect storm for emerging infectious diseases. And we do know that the climate change has affected diseases in the Arctic already. And this is just a little example, kind of the quintessential example of how climate change is affecting parasites in the Arctic. The muskox lungworm. This is work that was done by Pratap Kafli, a PhD student in my lab. And he showed that range expansion of this lungworm was caused by climate warming. In this picture on the far right, if we zoom into this muskox, you can see that it's got some bleeding out of its nose. And this is caused by this lungworm that we call Umingmux strongylus palicucensis. It lives in the lungs, forms nodules, and these animals can get infections of up to several hundred of these cysts in their lungs. And then when they're stressed, running, that will cause bleeding in the lungs and the subsequent bleeding in the nose. The worm requires slugs for larval development. So larvae are shed in the feces, they infect a slug. The temperature determines how fast those larvae in the feces develop to what we call L3, or the infective third stage, which muskoxen can then ingest. And this is really the temperature piece of this, and why the recent climate changes have really changed the distribution of this parasite. So when I started my scientific career, this lungworm was restricted to the area down on the mainland by Kugluktuk to the west, over to the Mackenzie River across muskox habitat. 2008, we saw that move up onto Victoria Island, and by 2017, it had made its way all the way up to Ulahukto. And the work that Pratap did showed that, in fact, this range expansion was very much linked to temperature. So you can see here on the left what the transmission index, so really what this represents, the colors, is whether or not the parasite could develop in a slug in a single season. And back in the 80s, it could just barely on the southern part of Victoria Island. And at that time, the lungworms were not present. So probably the seasons were just too short for it to take hold and get established there. If you look on the right-hand side, you see that in fact, in temperatures 2012 to 17, that transmission index has moved, is much greater on Victoria Island, and you see that the range has expanded up further north on the island. And all these yellow boxes, again, are where the parasite is present, the blue ones where it's absent. So certainly climate warming is affecting disease transmission in the north, and this is just one example that's being empirically studied. But beyond that, we are seeing more and more reports of unusual or increased and unexplained wildlife mortalities in the north. And the climate links to this we really haven't determined, but certainly we are seeing more reports. So Erysipelathrix ruseopathy, the bacteria that we're seeing in muskoxen on Ellesmere Island, has been reported elsewhere in the Arctic, causing widespread mortalities. We're seeing geese die in the thousands. There's been several events of that near Cambridge Bay, but also elsewhere. And then mysterious disease outbreaks killing seals in the Arctic. And a lot of the time, our challenges in understanding what's going on here are exactly the same as the challenges I articulated or illustrated with Ellesmere Island, and trying to understand what's causing animals to die there. Really, rarely can we get to samples in enough time, fast enough, so that we can get good tissues and good diagnostic samples. So that's the climate piece, but also emerging infectious diseases in wildlife can have a disproportionate impact on people in the north because it is a very vulnerable population. It's a lot of inequities, poverty, and food insecurity. So one figure here on the left just shows you the disparity between your income if you live in the north and if you're Indigenous versus non-Indigenous. This contributes to high rates of food insecurity for people in the north. And the figure on the right shows you the degree of food insecurity in Inuit Nunavut. So the first column on the left is for Nunavut households, and you see this dark gray color, 35% of people are severely food insecure in Nunavut, 35% moderately food insecure, and 30% are food secure. This compares to the far right, which is the Canadian average. So this is a huge issue. And healthy wildlife can really help to combat that food insecurity. A study by Woltier et al. In 2021 indicated that up to 5 million kilograms of country food are harvested per year in the north, and these are from 1996 to 2000. It's hard to say if that figure still remains, but at that figure, that food is valued at about $198 million food replacement value. And part of the reason this is such a high dollar value is because of the quality of the country foods. It's protein and energy packed, and therefore ranked very high on the financial component. But also healthy wildlife provide economic income. They provide income through tourism and through sale of handicrafts, and all that together can help combat the food insecurity. But healthy wildlife are also really important beyond this food and income. And I love this quote that came out of the Alaska Food Security report that, after all, our traditional foods are much more than calories or nutrients. They are a lifeline through our culture and reflect the health of the entire Arctic ecosystem. So the problems that we have then in the north, there may be a number, but around wildlife health, is that we are seeing increasing morbidity, so sickness and mortality in Arctic wildlife. We are seeing declining wildlife populations, and that's really illustrated by caribou population declines across most of the Arctic, with very few of them showing recovery. And we are seeing the emergence of zoonotic diseases. So these diseases that are in animals, but can also infect people. Erysipelathrix is one of those. Together, these things are affecting the health and sustainability of Arctic communities, Arctic cultures, and ecosystems. And ultimately, people in the north want to know, is this food safe to eat, and will there be wildlife here for generations to come? Complex problems like this require that we draw on all sources of knowledge. And this is a quote out of a report by the UN several years ago that says adaptation in the Arctic that integrates and respects local knowledge and Indigenous knowledge will be vital to help Arctic societies address the coming challenges. The IPCC also indicated that taking account of interdisciplinary scientific information, Indigenous knowledge, and local knowledge and practical expertise is essential to effective ecosystem-based adaptation. So essentially, we cannot just take what's been happening in the South and transport this to the North and say we can fix all these problems. We really need to be working with people in the North and using different knowledges to come together to try and address these problems. And so this is really the approach that our research group tries to take, a two-eyed seeing approach, with the philosophy that learning to see from one eye with the strengths of Indigenous knowledges and ways of knowing, and from the other eye with strengths of Western knowledges and ways of knowing and learning, to use both these eyes together for the benefit of all. And with that, I will I will spend the rest of my talk really going over the community-based Caribou and Muskox health surveillance program that our team has been doing over the last 15 years. So the program itself takes place primarily in 3 communities in Nunavut and Northwest Territories: Ulahukto, which is in Northwest Territories, the Inuvialuit settlement region, and Kuluktuk and Ekaluktutiuk. And the program itself is a very close partnership between the hunter and trappers organizations in these communities, the governments, and our university group. The philosophy around this is that we want to bring together Indigenous and local knowledge, targeted scientific studies, community-based surveillance to understand wildlife health, to detect changes in wildlife health and predict those changes in wildlife health, with the hope that we can improve wildlife conservation and stewardship, and also human health. So really a One Health approach, a One Health rationale for this. To do this, we've got 3 main components, I would say. We have interviews that are gathering local and traditional knowledge, and this includes baseline interviews in each of the communities, together with annual interviews to try to understand what's going on an annual basis. Targeted research and outbreak investigations, and then also a harvester-based sampling program. So I'll start with the interviews and how we document local and traditional knowledge. And really what we've been doing is using participatory appraisal techniques. And this was pioneered by Matilda Tomaselli, a former PhD student and somebody who now works with Polar Knowledge Canada. And what it entails is really using participatory methods to try and document the knowledge that community members have around wildlife health. Includes participatory mapping, drawing, timelines of events, seasonal calendars, and proportional piling, which is really using beans to quantify different indicators. Now, Matilda did a lot of work with the community of Ekaluktutiuk, or Cambridge Bay, in the mid-19 trying to understand muskox health, and ended up also understanding caribou health in doing this. But I'll present just a little bit of this work. One of the things she did first is document the area of observation that people were using. So what parts of the landscape were people using? And you can see on the left here, the green and different shades going right to orange that show the most heavily used areas of that her interviewees were using. She then asked people a whole variety of things, but one of the exercises was this drawing exercise where she said, in your lifetime, how have muskoxen been doing? And each of the different types of symbols in this graph on the right indicates a different group. So she had 7 different groups who documented in their lifetime what the trends for muskox populations were. And then she filled that in with a best fit curve here. And what you see is that there were very few muskoxen in the 60s. That increased to the late 1990s, early 2000s, and then there was a dramatic population decline. This decline, and her interviews were occurring in 2014. This decline that was reported by local and traditional knowledge really was also reinforced when surveys were done in 2014 that showed the population had declined from about 38,000 down to about 10,000 in 2014. Now, people did talk, it's not just about numbers, and I think that's been a really important lesson from her work, is it's the narrative that goes with those numbers. And one of the things that people talked about is the reasons for those declines were associated with vegetation, diseases, disturbance, and predators. So Matilda probed more on that and asked people about, well, how were muskoxen doing before the decline versus during this decline? And she probed into different things like body condition, causes of death, calving, a number of young animals, etc. And on the right-hand side here, I'll just illustrate a little bit of that. So body condition is the top graph, and you can see that pre the muskox decline, animals were generally in excellent to good condition, but during this decline, their body condition had changed to being good to fairly good to a lot that were, to more animals that were in poor condition. Their health status before the decline was pretty darn good, with very few diseased or dead animals. But during that decline, people saw a little bit more diseased animals and more dead animals. And when she pried into the cause of death on the bottom graph there, you see people said in the past, pre-decline, really the main cause of death was predation. But during the decline, the acute death had become more of a cause of death. So this animals found dead, suddenly dead. And then predation also had increased during that time period. She probed a little bit more into these deaths, these unusual mortalities or acute deaths, and asked people, okay, in this area that you are using heavily, what have you seen for dead animals? Before she did these interviews, we knew that there had been a mortality event for muskoxen in Wellington Bay, where 10 dead animals were reported in August of 2010. When she did the interviews and had people map out dead animals, she didn't generate, this is the knowledge that was there, she documented this epidemic curve where there had been actually a huge mortality event occurring over several years that had been completely missed by science, but was known in the community. So I think this is probably one of the most powerful illustrations of why and how traditional knowledge and participatory epidemiology techniques can be used to better understand wildlife health and be used for surveillance in the Arctic, in an area where all these other ways that we do surveillance just don't work. The information people gave her about these mortalities also mapped out spatially and temporally what was happening. So the top figure here, the little map, shows the area of observation. The map with the colors on it shows you by year where animals were found dead, and the size of the figures, the dots show you how many animals were found dead during that time. So top right is the epidemic curve, really. And then on the bottom right is the seasonality of the cases. And that's where people were seeing them primarily in the spring and the summer, a little bit in the fall. And so these were showing us patterns of mortality that really helped to try and understand what's going on better. Now, we were able to get samples from the animals in 2010 because those are the animals we knew about. And in fact, the cause of death for those animals, this bacterium Erysipelathrix ruseopathy, was showing up again. Now, just briefly, Erysipelathrix is what we identified in Ellesmere in 2021 and then in 2022 again, and also on Axel Heiberg Island. But we can see that this outbreak was happening 2009 to about 15 on Victoria Island. 2012 and 13, some dead animals were reported on Banks Island. In 2017, a cleanup crew was working on Prince Patrick Island, and they got a hold of us and said, we're finding some dead muskoxen. And Fabian Mavro went up there and found a dead muskoxen, dead Arctic fox, and dead prairie caribou, all of which were infected with the same Arctic clone. So while this looks kind of impressive on the map, there's a lot of spaces in between. We have no idea what's going on. And each of these incidents of reporting were really just coincidental or serendipitous. Highlighting how important it is to actually, as Matilda did, ask people what's going on and try to document that in a systematic manner. Now, Fabian Mavro is another researcher in my lab who's been working closely with community members in Ulahakto to also understand muskox health. He too used these methods of participatory epidemiology. He did timeline exercises, and this shows one interviewee's depiction of the population dynamics for muskoxen in the area. And that ultimately a number of different the graphs like this were put together to generate this population trend from 1950s to about 2017. He mapped out people's use of the landscape around Ulahokto, and then also did proportional piling exercises to look at how muskoxen were doing. And in this case, it's herd composition. So the percentage of young animals before the decline, after the decline, and then also based on based on data sheets from hunted animals. So you see before the decline, the proportion of young was higher. After the decline, it was lower. And then the observations from hunter-based sampling are even lower than that. Now, he also looked at things like group size. So he had two populations of muskoxen, one across Minto Inlet and others by Ulahokto. And using those proportional piling techniques, he showed that when the herd was bigger before the decline, herd size was larger, and when the herds declined, herd size was lower. And you see again, these are the summation of the multiple observations from the different interviewees. So starting to give us some indicators of if we see these lower herd sizes that people are reporting, does that, even though they cannot give us an absolute number of animals, does this become an indicator that maybe something's going on with the population? Now, the other thing that Fabian did while these interviews were being done, people started to report that there were unusual deaths in this area as well, swollen joints and eye disease. These are some of the unusual deaths, just the individual observations. He compiled those. And again, we see these larger circles in 2010 to 2009 to 14, showing large mortality event here, 2015, 2011. So there seemed to be something going on here, really probably between even 2007 to 15, where there was higher mortality, similar to what was going on in Cambridge Bay. Unfortunately, we've never been able to get samples from these animals. The interviews were done after the fact, and can't confirm that that was also Erysipelathrix. But he also had people reporting eye lesions. And what's really interesting about this, in this this time period of 2010 to about 16, people reported swollen puffy eyes, thick slime over eyes, walking in circles, walking by sound, and white eyes. These types of reports also came out of Cambridge Bay in that time period between 2010 and 14. And on Ellesmere Island, this is what we're seeing right now in 2022, right in the midst of this Erysipelathrix outbreak. We don't know what's causing this at this point in time, but it's an interesting finding that this seems to be consistent across these different time periods of across these different locations where Erysipelathrix outbreaks have been happening. And these observations really help to point us towards, well, the next line of research really has to try and figure out what's going on with these eyes, and is this associated with the mortalities? Okay, I'm going to run through some of the harvester-based sampling parts of this program now too. So all of that was really the interviews, the local traditional knowledge, and how that is really guiding a lot of the other research. So in these communities, we do work with harvester-based sampling, both for muskoxen and caribou. I'm going to focus on muskoxen because it's far too much to talk about otherwise. One of the key things that we've done when setting up these programs is had to really work closely with the harvesters to understand what is going to work for sampling. So what things can be sampled reliably, what tissues can be sampled reliably, what is acceptable to sample. Certainly there's some tissues that may be important culturally, they may be prime food sources, and having those discussions around that. Balances around the number of samples that a scientist may want versus the willingness to sample. And there's a tipping point there where people are going to say, no, that's too much. Environmental limitations, when it's really cold out, the last thing you want to be doing is things that take fine motor coordination, like putting small samples into small bags. And then also, are the samples that are collected, can we process these in the lab, and can we develop techniques that are well suited for working out in conditions like the -40, -50s, -60s? And this blood on filter paper has been one of those techniques that we've used. So rather than people trying to collect blood in glass vials, can they just dip filter paper into blood and put those into an envelope and dry them? So that's some of the work that we have done. And that's work that Olivia He has been doing just recently, trying to determine if we can use these blood strips to diagnose pregnancy in muskoxen. We know that it can be done with serum samples. So if people collect blood in tubes, we spin it down and get the serum, but can it be done with filter paper? And this graph here shows the correlation between filter paper blood strips and serum, showing that in fact, yes, these blood strips are accurately predicting pregnancy compared to the samples, the serum samples. This little blue guy down here that shows a pregnant animal is actually one that had just aborted. So it's a kind of a fuzzy zone right there. But this validation of techniques that can make it easy for hunters to do surveillance is really important. So some of the outcomes of this program have been sample kits. So the hunters go with the forms, they fill in those forms, which have various information about the animals they've harvested, but they also document body condition. And this shows you the back fat measurement over time. And this is number of animals harvested over time. And you see a very seasonal pattern, but you also see over time that it seems like there might be a slight increasing trend in body condition. Another way of assessing body condition is by hunters just saying, for this time of year, this animal is fat, really fat, not bad, or skinny. And again, we see the same sort of seasonal patterns and this slight increase. So again, these may be good indicators of how the animals are doing. From the kits, we get all sorts of different samples, and Indigenous knowledge in the interviews talked about animals being skinnier, and when the hunters sample animals, we can see also that's confirmed or triangulated with these samples. People talked about scabs on the mouth, nose, and here we have a sample from a muskox that had scabs on the nose, and with that sample we were able to test it and found out there was a new orthovirus in the animals. Hooves, people talked about overgrown hooves, and that's a project that's underway. People talked about tooth abnormalities, and I'll talk a bit more about that. And they talked about eye lesions. And then they also talked about fewer calves and limping animals. And I showed this picture on the bottom right is actually a bone that was submitted that we cut in half. The hunter hadn't actually noticed anything wrong, but when we cut that in half and saw the abscess in there, we cultured it and found out it was brucellosis. This became very interesting to us because brucellosis was not reported commonly in muskoxen. And Matilda Tomaselli then went and investigated this further. And in fact, this ended up leading to our realization that brucellosis had emerged in muskoxen on Victoria Island. Now, brucellosis is a disease that is known well in caribou in the Arctic. It can cause lameness, abortion, reduced fertility. Signs in caribou tend to be swollen joints, udder, mammary, and testicles, and you can see this big lump here on this caribou joint. And as I said, it's well known in caribou in the Arctic, but has been absent or very, very rare in muskoxen, and it can infect people. So we took these filter paper strips that we'd been collecting over years and years and started to test them for brucella. It was a disease that was not on our list initially, but because of these hunter observations and a few of these samples that came in, we thought we better test it. And what we see in Ulahukto is that this really has emerged. Up to 40% of the animals in 2021 that had been harvested by people and subsequently, the majority of which had been eaten, were exposed to brucellosis. Historically on Victoria Island, seroprevalence was less than 1%. Around Ekaluktutyuk, it's about 5%, but Ulahukto seems to have quite an emergence of that in that place. So some of the signs of it, again, people have recognized this disease in caribou for a long time, but in muskoxen, it's very new. And what we're finding is that people are not, hunters are not recognizing it as brucellosis, and subsequently they end up eating animals that are infected. And this just shows some of the things that have come in. This is an abscess in the meat on the top right. This is an abscessed lymph node on the left. The bottom left was a kind of a core, like a growth on the caribou's leg that just looked fairly benign. When we cut into that, it was full of pus and was also brucellosis. And then this is a kidney on the bottom that when we received a sample kit and started to process the kidney, we found some abscesses. So really, this is a syndrome that's shown up and is new to people. There doesn't seem to be a lot of traditional knowledge around it for muskoxen, and is a growing concern. We've had tons of communications back to the communities about this and to public health to ensure they are aware of it. Okay, just a couple more things here that I'll go through that are of interest, and there's tons more, but one of the most interesting things we've seen when we've been getting samples from these different communities is a really high rate of dental disease in muskoxen. And this is a figure put together by our graduate student, Erica Suter, looking at different dental abnormalities in muskoxen from Victoria Island and the adjacent mainland by Kugluktuk. And you see that there's teeth that are fractured, there's teeth that are cracked, have these horizontal cracks and vertical cracks, some really bizarre ones where teeth are rotating, some that are incredibly severe and others that are not quite as severe. Teeth that are displaced, so big gap here, and then teeth with this anterior wear, so wear on the front of the teeth. And then some asymmetry, so muskoxen should have 8 incisors, this one has 7. And this one has 9, so with extra teeth. So Eric's been looking at this, trying to understand what's going on. She's using not just this examination of the gross jaws, but also x-rays and CT scans to try and understand, why are these teeth growing in here so crooked? And is this in part because there's just not enough room in the jaws for them to grow? But also, are they fractured? Are there teeth under the gum line that are fractured, and are the teeth actually present? So for example, in this one, we've got a bunch that are fractured, 1, 2, and 3 on the in this middle figure. But also it seemed like it was missing its 4th incisor, but you see that on the radiograph. Erica's work in looking at that, she's compared the island and the mainland and seen very different types of syndromes happening. So on Victoria Island, muskoxen have really severe fracturing of their teeth. This first graph here on the left shows the muskoxen over 4 years old and really high rates of fracturing. The younger ones are not so high, but way higher rates than what's happening on the mainland. That unusual anterior wear, though, seems to be something that is happening only on the mainland animals and primarily in older animals, but also it's showing up in the younger ones. And so we have to wonder what's going on there. Are they scraping at things? What's different in the ecology of these animals that we're seeing these different types of pathology? And of course, this is, there's various causes and consequences of this incisor pathology. Causing wise, trace mineral imbalances may be an important piece here. And we do know from other work we've done that muskoxen on Victoria Island are deficient in key trace minerals like copper and selenium. Genetics can be a huge thing for dental growth. And we also know that muskoxen have the genetic diversity of a cheetah. And so maybe this is catching up with them. And when we're seeing these crowded mouths and rotated teeth, it might be associated with that. Vegetation type is changing. We know there's shrubification of the Arctic and that displacement of teeth, maybe that's associated with woodier shrubs getting stuck in there, but also is there increased mechanical strain with increasing icing events? And then what are the consequences of this? Does it cause increased susceptibility to disease, lower body condition, lower reproduction? And how is this playing in general health and population dynamics of these animals? So each of those samples that we take from the harvester-based program do get analyzed for a whole variety of different things, and that allow us to know a lot of different indicators of health. But we also, through the program, are finding that passive surveillance, so people just telling us that they've seen something abnormal or telling the wildlife officer that there's something abnormal there, is increasing. And that's both through direct contact, but also through social networks. And so frequently, Fabian at Kutz Lab is tagged on Facebook about these different abnormalities that are seen. And then, of course, the other social network is that Ronan knew Dean Clough, who knew me, and said, hey, there's something going on here on Ellesmere Island. Somebody should look into it. So various different ways that this information is coming. Last little piece here I want to mention is because I've talked about interviews and I've talked about sample-based surveillance, but the other thing that is really important in developing further with the Equinahodi K, or Boots on the Ground program with the Kutz, is that people are on the ground watching caribou. And this is also a way for monitoring and doing wildlife health surveillance. And my postdoc in my lab, Ben Padilla, has been working on that with the Kutz around this program to better understand how can we document from observations people on the ground, how can we document caribou health on the ground. He's worked with an undergrad student to develop some guides on how people can do this from based on observations, so remotely watching or non-invasive observations of caribou. So looking at things like body condition, strange walking, something strange around the head, something strange with hair coats. And they've put this into an actual guide and field notebook where the elders and monitors on the Boots on the Ground program can record these things. So where does this all go? So we're recording a ton of information, but really, how can we operationalize this information in wildlife stewardship and decision making? And I will just spend a couple minutes on that. If we look at the work being done in the Kutz, one of the key things is that we can start to link this caribou health information to other environmental factors, or we can link the muskox health information to other environmental factors to try to understand how these environmental factors may be influencing or associated with broken teeth, body condition, infectious disease status. So all of the data that are gathered through these programs, whether it be sample-based, observation-based, or interviews, can start to come together to understand the ecology of animals and some of the drivers of health. But we also want to use these in a more proactive way to try to get ahead of the game. So when population surveys are done, it's often too late, the herd's already in decline. But there may be early indicators of population health that can tell us that something is going to change. If body condition is getting poorer, if body size is smaller, if there's increased infectious disease, those may be early warning signs that can be used to guide wildlife stewardship, wildlife management, or even to stimulate a survey, population survey, to say, you know what, something's up here and this needs to be looked at. And this work was done by Stephanie Peacock, a postdoc in my lab, starting to think about a traffic light system where we can use these indicators to guide understanding the status of a herd. So on the bottom, I'm going to start at the bottom of this figure, and you see these different conservation status: red, amber, green, where red is the population's at risk, green is it's doing great. And really what we can do is we can go up to the top of the graph and say, okay, body condition is a really important indicator of health. Animals need to be in good body condition to survive. They need to be in good body condition to reproduce. We have a number of different ways of measuring body condition, so different metrics that vary by the knowledge source, so some scientific or local and traditional knowledge, and the way the body condition is tested. So captures, caribou captures might be provide a condition score. Hunter-based sampling can provide a back fat thickness, but also a hunter observation. And the relative condition can be documented through local and traditional knowledge interviews or annual interviews. Each of those may give us a certain target that says, hey, the animals are doing great for this season, if you remember these graphs that show you seasonal differences. Or they may say, man, this is a lot skinnier than they should be this time of year, and start to guide that, or start to provide some insights as to whether or not these populations are at risk. So to do all of this is obviously a huge amount of work, and it's nothing operates nearly as smoothly as I've made it sound throughout this talk. It really does require a lot of time on the ground, working closely with communities. I always remember one of my mentors saying that they'd sent coolers for samples up to the north and they were never sent back. And I kind of laugh at that, because they were put to really good use, I'm sure. But without people following up, without ongoing contact and training and interactions and feedback and getting jointly interpreting data, the coolers just aren't going to come back. And so we've done a lot of work looking at working with local wildlife monitors, spending time training them, doing wildlife disease workshops, lots of work with the youth, bringing these samples into the classrooms, really trying to support on the land camps that help not just the science harvester knowledge translation, but really the elder youth knowledge translation. And then projects that are co-designing, co-implemented, and co-analyzed. If we're not addressing the concerns of the communities, we're not doing things right. This figure here is just a quick one to show that we also had a project to evaluate how this all worked during COVID, when things were shut down, when we weren't any longer able to get to the communities and interact with the communities. And support them. And the program kept going, but certain shortfalls were identified, or vulnerabilities. And this is work colleagues of ours did to evaluate it, identifying the different components of the program, but also trying to find the ways to protect that. And I think that this is really neat work when we're thinking about doing these things long term. So with that, I'll just conclude in saying Arctic wildlife health surveillance is and needs to be multidimensional. Paper put out a few years ago about wildlife health surveillance in Canada suggested that health surveillance needed to extend beyond the physiological state and presence and absence of pathogens and toxins. And really that's what it has been focused on in the past, is it's surveillance for disease versus thinking about a broader definition of health. So it should include a broad range of metrics: behavior, habitat use, demography, population dynamics, body condition. And that's what we try to do with this program. It needs to incorporate different ways of knowing and ways of interacting with the land. It needs to recognize different communities interact with animals at different times of the year in different landscapes, and bring this complex ways of knowing caribou, these diverse ways of knowing caribou or muskoxen together to bring together a big picture. With that, I think I'll stop, and I would love to have some discussions, and happy to take questions.
Thank you, Susan. That was a fantastic talk, and I'm sure everyone is clapping in their own little space, saying thank you for giving that presentation. We have a couple questions here, so if it's all right, I'll just go ahead and read those out and get going with some of those. So Claudia Haas has a question here. She's actually my PhD student, and she is currently in Waterloo, but usually based in Yellowknife. So Claudia asks, were the carcasses left after samples were taken for species that died from, and I'm going to completely botch this name, Erythioplethorix?
Erythioplethorix.
Thank you. Is there a risk of infection of predators feeding on the carcasses, and is there a risk to human health?
Yeah, excellent question. Yes, the carcasses were left. There was no way of any alternate disposal of them, especially when you look at the remoteness, the number that were out there. But also in our absence of knowledge of this disease. Is it something that we need to worry about it being on the landscape or not? It was an area that has zero to almost no hunting in the area. It's quite a long distance from the community of Gris Fjord. That's the closest community, and so people were not immediately harvesting in that area at that time of year. But it does speak to the importance of understanding the diseases too. Is there a need to burn these carcasses, or are they better off left as is? The question of predators is really interesting because we actually have been collecting predator feces to see whether or not we can detect this bacteria in the predator feces, and I can't tell you the answer to that at this moment. It's top secret, but in general, what we find with predators is they generally seem to be more resistant to these diseases, especially scavengers. They seem to be able to eat off of these carcasses and not end up diseased in general. Having said that, Arctic foxes, we know that there is a syndrome of erysipelathrix in Arctic foxes, so they may be more vulnerable to it.
Okay, thank you. And I'll just encourage anyone to, if you have a question, don't hesitate to put it in the chat, and we'll slowly get to it. Thanks, Susan. Sarah here has a question as well, and she's calling in from Waterloo, Ontario. Are there any known consequences for the hunters eating the meat of the infected animals?
So Brucella and erysipelathrix are actually a major focus of our research right now, and we're working with a group in the UK with a CINUC grant, Canada Inuit Nunangat UK grant, with focus on erysipelathrix and Brucella to better understand the food safety concerns. Erysipelathrix, we know it mostly from domestic pigs. That's where we have most of our information. And in pigs, there can be a carrier state, but that carrier state means they're carrying it in their tonsils or lymph nodes, so places that people aren't going to be eating. The syndrome we're seeing in muskoxen is they are dropping dead very quickly. So they probably go from infection to death probably within 24 to 48 hours. So the likelihood that somebody harvesting muskoxen is going to hit that time frame is probably pretty low, and they do get very sick. So our guidance to people is to, don't hunt animals that look sick, and if you are ever in doubt, that it should be cooked well, and because cooking will kill this and brucellosis. So
And maybe I'll just expand on that a little bit. You talked about those two, but are there any other? My mind first goes to caribou and chronic wasting disease.
Yeah.
I don't even know if that
Sure.
is
Yeah.
but kind of
No.
broader.
Yeah. Yeah. So there's all sorts of, there's tapeworms that people will see in the meat. And there's lots of traditional knowledge around those things. People have seen it. These are normal things that they will see and their grandparents saw in caribou or muskoxen or fish and birds and marine mammals, but are safe to eat, or they have ways of dealing with it. So you cook it well, etc. And that's so important to realize, because the moment that you start saying, okay, it's got this parasite, don't eat it, you get meat wastage, and it's not necessary to. So some of these diseases, though, like Brucella, erysipelathrix, chronic wasting disease, do have potential human health consequences, and yet we have to balance risk of those of exposure, likelihood of exposure, with the high value of those country foods, not just for nutrition, but for cultural health and persistence in that. And so a lot of the work that we do is around supporting people in making good decisions around what they're seeing, and providing them some of the expertise around, can you identify if an animal has this, and what should you do, and facilitating those diagnoses. So making sure that pathway from, if somebody in Ulahokto finds something weird in their animal, that it gets sent as high speed as possible, down to a lab, and a diagnosis is made as quickly as possible, so they know whether or not it's safe to eat that meat, or yeah. So the better we can make the more efficient we can make that process, the more likely that the better we're doing at protecting people.
Right. Okay, thank you. There, the questions, I was gonna say the answers, the questions are just rolling in here, Susan. So it's just 4:30, so we have lots of time if you're able to take a few more. Okay, so Woodworth from Yellowknife has a question here. So Brad says, there are indications that muskox populations are increasing and expanding at and below the tree line in the Northwest Territories, as you and I have talked about in the past. Is this surprising from a disease ecology perspective, given climate change and your findings in the Arctic? And are southern populations of muskox exposed to more or less or the same or different diseases as northern
Yeah.
populations? Yeah.
Yeah. So this strain of Erysipelathrix that we see in the Arctic archipelago seems to be unique. And I think it is the unusual, it's the exception to the rule. And we think there may be something going on actually with that strain that is making it particularly pathogenic and virulent. We have detected Erysipelathrix in the odd case of muskoxen, moose, caribou on the mainland, and it is a totally different strain, and we don't seem to see these big mortality events. So that's the pathogen side of the host pathogen environment triad. Muskoxen in the south are exposed to many of the same things that they are in the north. One of the concerns I do have, especially with that East Arm population that keeps moving south, is exposure to bison. And that's going to be, muskoxen seem to be little pathogen pots. They invest in fat and good kiviot, and they don't invest in immune systems. So I think that's an issue. Is it surprising that they're increasing there while we're having these problems in the Arctic archipelago? I think the Arctic archipelago, like I say, I think it's the exception. I think Erysipelathrix up there is doing, and we're working on understanding this. It's got some different virulence factors. I think there's something going on there specific to the pathogen. Any disease is ultimately it's the culmination of host environment and pathogen, and it may be that the richness of the landscape in the south is compensating for a lot of these other problems and making animals more immune. But that's a whole long conversation for sure.
I'll just ask one question to build off of that. Is there evidence of inbreeding depression in either population?
Oh yeah, they are. Chris Kyle out at Trent University did some really nice work, his grad students. And I mean, I'm not a geneticist, but muskoxen have undergone huge bottlenecks, incredible bottlenecks. We sent tissues to him that we knew were from different animals. And he came back with 15 tissues from different animals we'd sent. He came back and told us we only had 10 animals. I think it's the equivalent, especially on the Arctic archipelago, Victoria Island area. It's the area we really focused on. Less diversity than a cheetah. The mainland's got a little bit more, but it's really bad. So huge inbreeding depression.
There's a couple more here. I will just bring up one from Alex as well, who just posted in the chat here. So Alex asks, and of course, Alex is based here in Waterloo, as you know. We often talk about two-eyed seeing from the perspective of Western researchers. Can you speak a bit about what you've heard from Indigenous knowledge holders about how the collaborations with your team have reinforced and reshaped their capacities as harvesters and producers of traditional food? Maybe do you want me to repeat the last part there? Can you speak a bit about what you've heard from Indigenous knowledge holders about how the collaborations with your team have reinforced and reshaped their capacities as harvesters and producers of traditional foods?
Right. Can I speak of how it's reshaped them? It's an interesting question because obviously the moment you touch something, you change things. And I think that there's no doubt that our interactions have been very two-way, and we have brought our scientific ways of knowing to the communities, while at the same time learning about the Indigenous ways of knowing. I think from the perspective of how has this reshaped, I just have to find that question.
You have it there?
No, I don't.
Oh, sorry, it's directly to me.
Have it. Yeah.
Alex, feel free to chime in.
Sure. Sorry,
Sure.
I didn't mean to throw a wrench in the works.
No,
It's a
no,
great
I didn't.
question.
I just was thinking, it's I mean, it's incredibly rich the way you're working with the communities and the two-eyed seeing approach, and how much you're working with that Indigenous and local knowledge, and it's clearly beneficial in terms of understanding what's going on. But often we approach this kind of understanding as non-Indigenous researchers. We're approaching it from that Western
Yeah.
researcher perspective. But I think that there are these really interesting transformations of what is Indigenous knowledge happening on the other side, because they're also going through this process of two-eyed seeing and embracing it. So I didn't mean my question to be a critique at all, but really, what do you hear your partners saying about how their interactions with you as a research team are kind of changing their own approach, especially as they're dealing with new diseases that they haven't seen before, so their existing knowledge maybe isn't serving them as well, so they're adapting.
That's just such an excellent question, Alex. And I think, and I would say, self-critiquing the work that I've presented today, it is very much come from the Western perspective of participatory epidemiology, gathering information, and then putting it into tables and figures that we understand as scientists. And I'll call it what it is. It is very true. That's what it's doing. And in a way, it's a knowledge translation. It's one of those steps of translating the observations that people have to something that we as scientists are used to dealing with. But it's not the end point, for sure. And it's not the only way, and it may not be the right way. Other half of my research group is working very closely around Indigenous knowledge through very deep and extensive interviews, and delving much deeper into a lot of the relationships that people have with animals, the interactions, and it's sometimes mind-boggling, I think, as you get deeper and deeper into that. So I think it's really, really important to respect that. It's really important to maintain the narratives that are happening with the participatory research. Because if you just take the numbers and figures without the explanations, then it becomes it's not accurate, it's not right. Everything that I've presented has gone back to the communities for that interaction and make sure it's correct. But I don't think, so I don't think it's changing the ways of people. Just coming back to that question, is it changing? But I think what it's doing is it's building really good relationships and trusting linkages between the scientists and the communities. And I think it's supporting collaborative wildlife stewardship. Because the data that we've got have been generated by the communities, whether it's traditional knowledge or samples, and our obligation is to them first, not to the government first. Sorry, government folks, but you're aware of that. But it's to the communities first. And I think what that means when it's working well is that then the communities can go and say, we trust this information. It's come from us. And it's a stronger starting point when sitting at the co-management table.
Thank you, Susan. Here we have Kira. Kira is also calling in from Waterloo here. She says, I may have missed it, but does lungworm have the ability to stay in the infected carcass for a long period of time after the animal dies? If so, would this period of time increase with higher global temperatures?
So the lungworm actually it's an amazing little creature. Those first stage larvae that are shed in the feces can survive -40. Actually, they can survive -80 for probably decades. They can survive desiccation. They're probably not as good at surviving heat. the lungworms in the carcass themselves are not infective, and the adults would die. They'll die with the animal. It's just those larvae that are passed in the feces that are really tough and can survive.
Okay, thank you. Yeah. I was going to
Yeah.
say, speaking of government, we have a question from Dean Clough here. Dean, I presume you're calling in from NWT. Dean asks, are you planning a trip to Ellesmere Island again this summer, or do you wait for field reports?
Yes. So 2021 was the first year up there at the last minute and investigate the carcasses. Last year we went up and we did follow-up studies on the carcasses that we had seen, on carcasses we hadn't seen but knew were there, and any new carcasses. Government of Nunavut at the same time did some helicopter surveys or helicopter reconnaissance to see if the mortality event was still happening, and it was. And they also went to Axel Heiberg and found animals there and actually saw animals dying, which was sad, but also got us fabulous samples, which really allowed us to understand the pathophysiology of this better. We are going back up this year. Our plan is to resample carcasses that we went to last year. So we, and the year before. So we are doing that. If there's bones there, we take those bones, resample them. We also are sampling the soil to determine whether or not the bacteria is persistent in the soil. We're sampling bugs, we're sampling rodents, to really, and any other species we can, to understand how is this being maintained, how long does it persist in the soil, how long does it persist in the environment, and also to try and understand these eye lesions. So yes, we're going back, and it's amazing. We learned a lot from the outbreaks on Victoria Island and elsewhere that have allowed us to say, this is really important. We're probably at the start of this outbreak, and it's really important to track it now in as real time as we can to better understand it.
And maybe apply to other, some of the mainland populations that are increasing, or apply to other areas too. Yeah. Okay, so the next few questions are from my fourth year students, which is great to see. So all calling in from Waterloo. Matt asks, are more coolers coming back now? How has outreach to Indigenous communities been received, and are local communities in the NWT and younger generations more engaged?
Yeah, more coolers are coming down from the communities we're working in. And there's a little bit word of mouth that happens that extends out to other communities. There is ongoing surveillance and support both in both the territorial governments. So I think it's important that anything that does come in, those unusual observations always go through territorial governments. They may end up in our lab for diagnostics, but they always should. There is a system in place there in the territorial governments. I think the presence that we've had in the community with the increased knowledge about disease and increased knowledge that that something can be done, things can be tested, has increased the submissions for sure. Yeah. Is there? Yeah.
Yeah, there's a few more, Susan, if you have have the stamina and time.
Absolutely. No, I'm happy to.
Thank you. Ethan, also from Waterloo, asks, is there a correlation between the time of year / season and higher levels of infected individuals?
The best knowledge we've got is from that graph I showed you with from Eklututiak, where people describe the seasonality for erysipelothrics, I think is what your question is about. So it seems to be most common in the summer based on that information, and based on the real surge of die-offs that we saw on Ellesmere. There didn't seem to be a bunch of carcasses there when the film crew arrived, but that hot period of the summer, and they did report extremely high heat and mosquitoes like you wouldn't believe. So there may be some factors there that are triggering this.
Coming together. Abby, also here from Waterloo, asks, if a female or male muskox is infected, could the infection be passed on to a child through either birth or the pregnancy?
Yeah, so these are things we don't know for muskoxen. We know for sheep that bacteria can cause abortion in sheep.
Okay. Madeline here from Waterloo as well asks, well, first of all says, thank you for a great talk. Does your lab plan to expand the community-based wildlife surveillance program to other communities?
Love to, but we need to multiply ourselves. I think my hope is that what we've done over the last 10 years with these communities has really demonstrated the value of it, and that we can help implement it elsewhere and start to turn it over from just being a Kutz research group coordinated thing to a broader surveillance system around the Arctic.
Good note to all the students on the call that we need more people doing northern wildlife work. So that's great. Lauren from Waterloo here as well says, the eyes of the affected bison reminded me of the eyes of cattle who suffer from mad cow disease. Do you think that some of the bison could have suffered from this disease during this study?
I think you mean muskox, probably.
So, yeah.
No, I don't think at all that it's associated with that. No evidence and no reason mad cow disease would be up there. And even with chronic wasting disease, which has been tracked so carefully, it's thankfully still not up there. The eyes are, there is some sort of infection, whether it be viral or bacterial, or maybe it is erysipelathrix, hard to know. Yeah.
Okay, two more questions came in here. Steve Anderson asks, so you mentioned that erysipelathrix, am I getting that right now? Thank you, I'm learning. Is found as far west as the Mackenzie Delta. Do you know if it's also present in the muskox population to the west of the Delta, so in the Richardson's Mountains and a North Slope?
So we actually know quite a bit about erysipelathrix just in general. So the bacterial, the species or Ruseopathy, we have found that in muskox and actually in Alaska and in caribou, in Alberta and in Northwest Territories. And I don't know that we've found anything in the Yukon. We've found erysipelathrix in muskox and in the Sawtooth. But all of the strains that we are finding in those different places are not the same as what we see in the Arctic archipelago. And erysipelathrix is known to be, so we call it a generalist and an opportunist. So generalist means it can infect everything from people to fish and birds and mammals of all sorts. Opportunist means that usually what it does is it infects these different animals when they are immunocompromised for some reason, whether it be a wound that it can penetrate through the skin and get access to the body, or just the immune system not functioning well. So it's not entirely surprising to see it in other species, and we do see it in these other areas. And we also know it's present based on serology. So we've run blood tests to see if there's evidence of exposure in caribou and muskox. And so we know across the areas we've looked that it has been present and circulated, and there's animals that have recovered from it because they're showing antibodies. But the strain that we see in the Arctic archipelago causing these mortalities, we have only found that strain in that area. Everywhere else where we've sequenced isolates, they are a whole variety of different isolates.
Wow, okay, that's pretty fascinating. Okay, two more questions here. Paula is calling in from Vancouver. So Paula, she says, thank you, Susan, and your teams and all who are involved in protecting the Great North.
Thank you, Paula.
And Dylan, calling in from Waterloo here, says, you may have already covered this, but do infected carcasses have the potential to infect animal species which happen to eat the meat?
Yeah. So absolutely. We know because we take these carcasses and we bring them pieces back home, and we put them in a petri dish and we culture the bacteria. We know there's live bacteria in those carcasses. Most of the time, predators tend to be more resistant to these types of bacteria, or generally seem to be more resistant. So yeah, there's a big but of we really don't know for sure, and we do know that Arctic foxes can get this. So there's probably a mixture of things happening there.
Okay, well, that's it for the questions that have been coming in, Susan, unless anyone has a final one, but you've been on the spot for a long time. So I want to thank you very much. It's fantastic to have the time with you, and it's been a real privilege to be hosting you today and to be part of this webinar. So I'm sure everyone is joining me and giving a round of applause and thanking you very much for being with us today.
Thank you very much for the invitation, and thank you for the great questions out there.